Okay, so today we're talking about protein structure. This is just something I want to talk about real quickly because it's going to lead into our enzymes that we're getting into. Okay, and so proteins, I'm going to write a word and then I'm going to draw a shape. So the primary structure of a protein is a chain. It's a chain of amino acids. And so it's a chain. So if this ball is an amino acid, oh, excuse me, all better. So we have a chain of amino acids. So just a brief overview. We have our DNA, right? And every three nitrogenous bases is called a codon, and that codes for one amino acid. And going back to our macromolecules, for proteins, if protein is the polymer, then the monomer is the amino acid. So the primary structure of a protein is just a chain of amino acids. We call this chain a polypeptide. And they are made with little bonds that bond them together are called peptide bonds. I know I didn't spell peptide right. And also remember, so I'm going to doodle up here, that a protein just has an N, CC, right? So this would be a protein. They have some stuff going on up here, and then they have the R group down there, and boop. This would be the peptide bond, and then to another NCC thing, and then bap, bap, bap. NCC with an R, with your R. Okay, so pay attention to that. Secondary structure, we have our alpha helix. See, how do I draw that? Like an alpha helix. And beta pleated sheets. So I'm going to draw this as just one of these spiral looking things, and like it looks like a kind of like a wave, almost like just kind of jagged and connected. Okay, and so these are just made with hydrogen bonds. Hydrogen bonds mainly, and that's really all that's involved. Tertiary is it starts to folding together of the folding, like it folds together. So I'm gonna write folding. It's like a glob of protein from one chain out of these alpha helix and beta pleated sheets. And also understand that like alpha, you have an alpha helix and like a beta helix, and basically the difference is one turns one way and one turns the other way. Tertiary, I just think is like a chain folding. It's still the one chain, but I think of it as like a glob, because it's just all these shapes and pleated sheets put together, but it's still one chain. And then quaternary would be like multiple globs. Oh, and so in here you're also going to have R group interactions. So you may get some sulfur bonding, you may get some hydrophobic interactions and things like that. And then quaternary is just like multiple globs of protein. So multiple globs. That's how I think of it. And that's kind of an overview of protein structure. I tried to go a little bit in depth, but yeah, there you go. And thanks for watching.